Hello grade 12, how are you? Today you will start a new project in Omni International School as we are suffering from epidemic viral infection coronavirus. We will continue chapter 23 biological compounds. We finished describing the basic structure of amino acids and the formation of polypeptides, and we understand what's the meaning of polypeptides. Also, we describe the functions of proteins and cells. Today, we will start to determine the significance of amino acid side chain and the three-dimensional structure of a protein and the function of proteins. Before starting, we have to understand some facts. The first one, amino acid molecules are the basic building block of proteins. Second, more than 700 types of amino acid occur in nature. Third, only 20 types of amino acid are found in human protein. Fourth, the human body can synthesize only 11 of the 20 amino acids as needed, and these 11 amino acids called non-essential amino acids. The other 9 are called essential amino acids. This is the basic structure of amino acid. It consists of carboxylic group, functional group, amino group as a functional group, and central carbon, and this central carbon is connected with R group, and this R group determines the properties and functions of amino acid or proteins. Long chain of amino acids are called polypeptides, but more than 50 amino acids are called proteins. Peptide bond can be broken or hydrolyzed by enzymes called protease enzymes. Each peptide, polypeptide, or protein is made up of a special sequence of amino acids. We can mention to the amino acid by the first 3 letters of the word. Polypeptides and protein function depend not only on the kinds and number of amino acids, but also on their order, and this order can affect the function and the genetic variation of the proteins. The properties of amino acids and ultimately polypeptides and proteins depend on the properties of the side chain present. Like in glutamic acid, it is an acid. Why? Because it has more than one carboxylic group, which is acidic in nature. The side chain of histidine is basic. Why? Because it contains amino groups. The side chain of asparagine and several other amino acids are polar. Why? Because it forms many of hydrogen bonds. Some amino acid side chains can form ionic or covalent bonds with other side chains, like cysteine, which has sulfide group which form covalent disulfide bridge. See this structure. This disulfide bridge, which is covalent bond, and you understand the difference between covalent bond and hydrogen bond. As mentioned before, we have 2 types of chemical bonding. We have intermolecular force, which are hydrogen bond, van der Waals force, and dipole-dipole force. The intermolecular bonds like covalent bonds, ionic bonds, and metallic bonds. Here we are related to the intermolecular force, which is between molecules. Here we can see the disulfide bridge between a molecule of cysteine and another molecule of cysteine. Also here, we have hydrogen bond, which is intermolecular force between a molecule of glutamic acid and a molecule of asparagine. And this type of bond gives the polarity property of the compound. Between these two types of bond, we have hydrophobic environment. What's the meaning of hydrophobic environment? Hydrophobic is composed of two parts. The first one is the prefix, which reflects hydro means water. Phobic means dislike. So this area dislike water. Why? Because it is non-polar area. So the interaction of amino acid side chains determines the shape and the structure of protein. These are important to the protein's biological function. In a polypeptide chain or protein, the sequence of the amino acid is called the primary structure, which is straight structure. The secondary structure describes how the chain is coiled or otherwise arranged in space. We have two types of secondary. The first one is alpha helix. The second one is beta plated sheet. A protein's characteristic three-dimensional shape is called its tertiary structure. Side chain interaction at various positions along the protein backbone cause the tertiary structure. The side chain interaction can include the hydrogen bond, which is the polar bond, salt bridge, which is also polar bond or polar bridge, cysteine disulfide bond, which is covalent bond, and hydrophobic interaction between non-polar side chain. Non-polar side groups tends to be found in the interior of the protein, where contact with water is minimal because it is non-polar, so it dislikes water. Polar and ionic side chain tend to be on the protein surface where they are in the contact with water. So here return to the diphospholipid bilayer and the cell membrane of the cells and compare between the two structures. As we know, hemoglobin is a type of protein. It has 4 different polypeptides make up the quaternary structure. So hemoglobin is quaternary protein structure. This slide explains the 4 structure levels of proteins. The first one is primary, the second one is secondary structure, and this is the tertiary structure, and the last one is the quaternary structure, which is hemoglobin. Amino acid substitution. A single substitution of one amino acid for another can change the shape and the function of the protein. The genetic disease sickle cell anemia can happen when glutamic acid is replaced by valine. And we understand inside the class this substitution action will change the shape of the red blood cells from the double concave structure, which is the normal structure, to sickle structure, which will affect the capacity of hemoglobin to carry oxygen to our cells. So now we understand the 4 levels of the protein structure. We have primary, secondary, tertiary, and quaternary. So today we finished our section. Thank you. See you. Bye.